Hello, everybody. My name is Thomas Durkin, and I'm very delighted to actually be getting invited by ODIN to present the Neuro's Early Drug Discovery Unit, or EDU for short, and kind of talking a bit about our first step from a target to a therapy. So, I'm an associate professor at McGill University, and I'm director of the Neuro's EDDU, which is based out in Montreal, Quebec, Canada. So, just to shows you where we are here, and it just shows the different types of seasons we actually experience. So, what I really want to talk to you today about is actually how we can help you as part of our first step program within the EDDU, how we can help you go from a target. You might have a gene and microRNA or something, and you want to understand what does it do? Is it a disease? Where is it in the cell? What is it even doing within the cell? So we can help you really profile it deeply to then figure out, is this an actionable target? And then maybe there's actually a disease-associated phenotype when something's perturbed. Maybe a mutation is added. Maybe you have a patient line where you already have the mutation, the gene gets nucleated. Once we have a disease-associated phenotype, we can then work with the groups, either an academic group, company, foundation, and families to then help potentially screen for candidates to eventually get to a lead. And really what our goal is then to see is how can we actually then help you with preclinical development, trials, and even maybe just at the end to have initial data that you can then go on to build kind of bigger, more kind of larger scale applications to bring your program to the next step. So what we have as part of our group is we really have a whole integrated system from the clinic through our group all the way back into the clinic. So we're downstream of what we call the Neuros Biobank or CBIG, where patients are consented in an open framework where we can collect these cells, we program them into iPSCs, we can correct them through gene editing, and then eventually then do the QC to build a cohort of iPSCs we can actually apply to make different disease relevant cell types 2D models, 3D organoids, to really build these assays, both high content, automated, all the way up to different functional activities, with the real goal to identify lead candidates to eventually go back into the patients that we actually see within the clinics in our hospital. So our pipeline, we work with groups anywhere from like novices that never worked at iPSCs. We actually have training and everything involved. You can do it virtually, in person. It's actually helping groups make their own iPSCs, CRISPR editing, making sure they're the highest QC, to even working with them to build maybe a new assay you haven't thought about, figuring out a new protocol to build a cell type you actually want to do within your actual workflows. We can help with different diseases of the brain, from Parkinson's to ALS to rare disease. Our pipeline really is agnostic, so we can really work with you depending on the idea and whether we have the expertise to help. We've over the last 11 years in existence, we've built a team of about 25 plus staff scientists and associates, assistants that really have the technology and expertise that can cover all these different types of projects. We've worked with many users, both within academia, industry, who published many different papers across these disease areas. And these are just the cell types we can actually make with established workflows from different types of neurons to glial cells, spheroids, organoids. We can even go and make new different cell types. We're in the process of making retinal ganglion cells at the moment. So if you even have a cell type that's not on the list, we can even help you work with it to develop that for the project. So how can we help you? Well, we can help you really develop disease-relevant iPSCs or even control iPSCs that you may need for your project needs. We can help you find a disease signature that maybe you haven't found before. And then once you have it, to then see could we then apply it to therapeutic testing to explore the use of these novel technologies with iPSC models. You might have a new way to functionally profile a neuron. A glial cell, for instance, we can help you then see can you get the proof of concept data and build it into something larger, all with the goal to test candidate therapeutics to really meet the unmet need that's out there today for CNS disorders. So what we do have is we have direct access to the iPSCs, to the expertise in-house infrastructure that we can help with early discovery testing. This shows you what we have: high content imaging, flow cytometry, single cell phenotyping, functional MAA, you name it, we can do it, even cytokine profiling. We have access to a diverse patient cohort from across within Montreal to across our province and even in Canada, and a clinical trial infrastructure to help move things forward if needed. And we do have strategic partnerships with KOLs across different CNS disorders, if need be, from patient advocates to foundations that maybe we can help bring into these different types of projects. So hopefully you enjoyed learning a little bit about us. This is just a snapshot. And if you want to learn more about us, I encourage you to check out our LinkedIn, Instagram. We have our iPSC catalog or different websites. And thank you for taking the time to listen. Take care. Bye bye.